The main advantage of this technique is that you can look at the cells real time in the automatic optical microscope. The implication of this treatment extends to a therapy of chronic wounds or cancer, where microgration plays a crucial role in these diseases.
Begin by washing a confluent culture of HACAT cells 2 times with 5 milliliters of sterile magnesium and calcium-free PBS per wash. Next, incubate the cells with 1 milliliter of trypsin in a 37 degrees Celsius incubator for 5 to 8 minutes. After confirming detachment under a light microscope, stop the enzymatic reaction with 9 milliliters of culture medium supplemented with FBS. And dilute the cells to a 7.5 times 10 to the 4th cells per 250 microliters of medium plus FBS concentration. Next, seed 250 microliters of cells to each well of a round-bottomed 48-well tissue culture plate. And gently slide the plate from side to side to ensure that the cells are equally distributed across the well bottoms. Then transfer the plate to a cell culture incubator. When the cultures have reached a 90 to 95% confluency, replace the supernatant in each well with